The human cell is an amazing structure which is busy carrying out various life-sustaining processes, one of which is protein synthesis. For this process, it is essential that amino acids which lie scattered in the cytoplasm are carried to the messenger RNA or the mRNA template, the site of protein synthesis. Till the 1950s, scientists did not know enough about how proteins were carried to the mRNA. The breakthrough came in 1955, when Francis Crick, who had earlier revealed the DNA structure and its properties, postulated the adapter hypothesis theory, in which he stated that it is therefore a natural hypothesis that the amino acid is carried to the template by an adapter molecule, and that the adapter is the part which actually fits onto the RNA. In its simplest form, this hypothesis would require 20 adapters, one for each amino acid. Soon after Crick published his hypothesis, scientists across the world worked to find this mysterious adapter molecule that adapts itself to carrying amino acid and reading it to the codon of mRNA. Success, however, came to Robert Hawley when in 1965 he discovered transfer RNA or tRNA, the adapter molecule, and also worked out its entire chemical structure. Today we know that tRNA, the smallest of all RNA molecules, is made up of about 75 nucleotides. Synthesized from a DNA molecule in a process similar to the synthesis of mRNA, tRNA is soluble in 1 molar NaCl and is hence known as soluble RNA or sRNA. Each tRNA has G at its 5' end and CCA at its 3' end. Interestingly, tRNA takes the shape of a clover leaf when examined as a two-dimensional structure, whereas it looks like an inverted L-shaped molecule in a three-dimensional structure. Regardless of its dimensions, a tRNA consists of 4 arms or sites: ribosome recognition site, anticodon site, enzyme recognition site, and amino acid binding site or carrier end. Each of the tRNA regions perform a particular function. Take the case of the ribosome recognition site, which recognizes specific ribosomes attached to the mRNA and brings amino acids to the ribosome for protein synthesis. On the other hand, the anticodon site consists of 3 nitrogen bases called the anticodon. During protein synthesis, the anticodon pairs with the codon of the mRNA strand. However, the anticodon of tRNA is complementary to the codon of the mRNA strand. Apart from the anticodon site, a tRNA molecule also consists of an enzyme recognition site, also known as the D-arm or dihydrouracil loop. This particular site is the recognition site for amino acid synthetase enzyme, which helps the amino acid bind to the tRNA. Incidentally, this binding takes place at the 3' end of the amino acid binding site. Did you know that there is a specific tRNA for each of the 20 amino acids found in the human body? Also, there is a specific tRNA called initiator tRNA, which initiates the process of protein synthesis. However, there are no tRNAs for stop codons. tRNA is thus an important molecule, and it plays a vital role in protein synthesis by bringing amino acids to the mRNA template in the cytoplasm. Proteins play a pivotal role in the proper functioning of any organism. The synthesis of proteins occurs in a cell in two phases, namely transcription and translation. The process of converting mRNA codon sequences into an amino acid polypeptide chain is called translation. Prior to translation, mRNA, which is synthesized in the nucleus of a eukaryote cell, migrates through the nuclear pore into the cytoplasm of the cell. The cytoplasm also contains tRNA, amino acids, and ribosomes, all of which are involved in the process of translation. The amino acids and tRNAs in cytoplasm become activated by a process called the charging or aminoacylation. During this process, amino acid binds with ATP in the presence of aminoacyl RNA synthetase and is activated into aminoacyl AMP enzyme complex. This complex reacts with tRNA and transfers the amino acid to it. As a result, tRNA becomes activated as aminoacyl tRNA, and the enzyme and AMP are liberated. The tRNA has an anticodon loop, which determines the amino acid that will link to it. This anticodon of tRNA is complementary to the codon sequence on the mRNA and will only bind to that codon. Apart from tRNA, a ribosome is another component required for translation. It is made up of structural RNAs and about 80 different proteins. In the inactive state, a ribosome consists of two subunits, a large subunit and a small subunit. The large subunit consists of two sites, the P or peptidyl site, also called the donor site, and A or aminoacyl site, also called the acceptor site. At these sites, the two subsequent aminoacyl tRNAs bind and facilitate the formation of a peptide bond between the two amino acids. The ribosome also acts as a catalyst in the formation of the peptide bond. Another component in translation is mRNA, which consists of the start codon AUG that codes for methionine, the stop codon, which is either UAA, UAG, or UGA, a region that codes for the polypeptides, and regions that are not translated into proteins called untranslated regions or UTR. These regions are present at both the 5' end before the start codon and the 3' end after the stop codon, and ensure efficient translation. Translation occurs in 3 phases: initiation, elongation, and termination. In the initiation phase, the small subunit of the ribosome moves along the mRNA strand to locate and bind to the start codon AUG. The first aminoacyl tRNA carrying the amino acid methionine with an anticodon UAC binds to the start codon at P site, after which the large ribosomal subunit binds to the small ribosomal subunit containing the mRNA. Another aminoacyl tRNA now binds to the A site of the large ribosomal subunit, and the first peptide bond is formed between these amino acids. The peptide bond formation is catalyzed by the peptidyl transferase enzyme. The formation of this bond requires energy, which is obtained from ATP. Simultaneously, the first peptidyl tRNA gets removed, leaving the amino acid. The next phase is elongation, in which the ribosome runs along the mRNA in the 5' to 3' direction, moving from codon to codon and adding subsequent amino acids to the growing polypeptide chain. This is caused by the elongation factor translocase, and this process is called translocation step. On reaching the stop codon, UAA, a release factor or RF, binds to it, terminating the translation and releasing the polypeptide chain from the ribosome into cytoplasm. After the termination, the small and large subunits of the ribosome dissociate and the mRNA degenerates. The polypeptide chain is therefore an end product of the translation process that takes place in the cytoplasm.